This is Amalthea. Even though the Human Development Index in Western society is high and people have access to enough food, there is an unprecedented rise in diet-related non-communicable diseases, or NCDs in short.
Malnutrition, simply put, is bad nutrition. Bad nutrition because you're eating either too much food or too little food. Too little food because you are not meeting your body's nutrient requirements, and so the body's growth and development is affected.
According to WHO, unhealthy diets account for a large share of burden in diet-related non-communicable diseases in Europe. For example, an estimated 86% of deaths and 77% of diseases. Those issues are well documented, and there are many efforts from governments and corporations to overcome them. However, a holistic and sustainable approach does not yet exist. We believe that we found the missing link to overcome them. The gut microbiome harbors trillions of microorganisms and plays a critical role in the integrity of intestinal barrier, metabolic homeostasis, maturity of the immune system, especially at the early stage of life. Being a rising field of interest, but still at its infancy, scientific information is still very limited. Among others, the bacterial communities interact with each other with short-chain fatty acids, one of the most abundant metabolites of the gut microbiome. When the levels of SCFA drops, the stability of gut microbiome flickers, a phenomenon called dysbiosis. This phenomenon is characterized by shifts in the abundance of bacterial communities, and their metabolic capabilities are decreased. This sets the prelude to diet-related non-communicable diseases. After interactions with several stakeholders, we decided to use SCFAs as our indicator for the evaluation of the gut microbiome. And this is where we step in. Amalthea, our synthetic biology solution to unlock the full potential of gut microbiome and to tackle diet-related non-communicable diseases. We have divided our project into 3 systems. First, we detect the shortages of SCFAs with the usage of a bioelectronic capsule. Then, the results are transmitted to a smartphone and an application where a team of multidisciplinary experts are evaluating them. Finally, if dysbiosis is in fact detected, we ameliorate it with a live biotherapeutic symbiotic supplement that produces the amount of SCFAs directly in the gut.
Our first task was to detect the absence of SCFAs, so we created a whole cell biosensor hosted inside an ingestible capsule, which consists of the metabolite detection module and utilizes a NOT-gate device. We tested 2 alternative metabolite detection modules for SCFAs, a GPCR-based and a module based on promoter flick. The GPCR-based detection module is activated in the presence of SCFAs. After its activation, recruitment of beta-arrestin occurs, which carries a TIV protease that cleaves and releases a transcription factor, inhibiting the reporter module.
We began by assembling the capsule and its components one by one, gradually upgrading it along the way. We chose a system-on-chip processor after we discussed it with Dr. Stόχος, instead of the basic microprocessor from our first iteration. This SoC is smaller in dimensions and is equipped with a Bluetooth antenna that can transmit the encrypted data to a mobile device. We also added another battery unit, thus improving both the safety and the lifespan of our system. Additionally, we designed a 3-electrode detection sensor, a choice confirmed by various scientists and stakeholders who also confirmed our choice of semi-permeable membrane. Finally, as a part of our kill switch mechanisms, we designed a reservoir to contain L-arabinose, which in case of an emergency physically communicates with the modified bacteria and then eliminates them. In order to proceed with our wet lab experiments for the whole cell biosensor, we performed in silico simulations. we developed a functional mathematical model to simulate the function of all our modules. Notably, we discovered how the GPCR module responds to the beta-arrestin 2 receptor concentration ratio and how that affects the signal stability.
Based on the in silico simulation results, we tested the whole cell biosensor in the lab, using a fluorescent protein as an output. The top and bottom lines in the graph represent the positive and negative controls, which mark free fluorescent protein and empty vector. The rest of the lines in between are GPCR with various concentrations of SCFAs. The expected results should have the line without SCFAs correspond to higher fluorescence, close to the positive control. There was continuous non-specific suppression both in the presence and absence of SCFAs. We hypothesize that 1. Either suppression takes place without the release of the lac from the GPCR before it gets to the membrane, or 2. The TIV protease cleaves the lac non-specifically. These experiments will be performed in the next design, build, test, learn cycle. Furthermore, to prevent an unlikely contamination event inside and outside of the body, we designed two different kill switches. The first one was based on temperature, which would kill the bacteria after capsule disposal. But after discussing with an expert in environmental microbiology, we replaced it with an auxotrophy-based kill switch. The second one is an arabinose-inducible kill switch that is mechanically activated in case the capsule breaks inside the gut. Regarding the reporter module, we utilized the enzyme tyrosinase, which converts tyrosine to L-DOPA and L-DOPA quinone, thus creating a measurable electrical current. Our last year's failed attempt on the tyrosinase assay was due to the use of a constitutive promoter. After several iterations, this year we managed to produce the enzyme by implementing an arabinose-inducible promoter, as you can see in this SDS page. To summarize, our bioelectronic capsule provides a snapshot on a patient's gut SCFA levels. But how can we evaluate this precious data?
Radical changes in the national health systems attempt to ensure better access to personal health data both for patients and experts, enabling faster diagnosis and improved monitoring. The idea of e-health was promoted even more throughout the last 2 years due to the COVID-19 pandemic. This is why we developed eAmalthia, a digital application that receives data from a biocapsule via Bluetooth. The exploitation of a bioelectronic interface enables real-time monitoring on the user's smartphone. eAmalthia was developed like any other app you can download on your phone. First, the user must log in. The same login applies both for the user and the expert that evaluates the results. This is the main interface of the app. Here you can see how the capsule's results will look like. They come into a concentration vs time diagram that represents roughly the real-time concentrations of SCFAs in the gut. Now, if this biosis is detected, we move on to ameliorating that phenomenon. Diet plays a major role in strengthening the gut microbiota's health, and this is why we provide a personalized dietary pattern. This diet is based on the user's data after being evaluated by a nutritional expert. Finally, both users' data and experts' opinions are well protected in this digital environment following the GDPR regulations about privacy and personal data.
After detecting and evaluating the levels of SCFAs to the patient's gut, we need a robust platform to deliver those molecules to the patient. To this end, we created a symbiotic supplement that consists of 2 parts, a probiotic able to produce the missing amounts of SCFAs and a prebiotic which feeds the natural gut microflora. We managed to, firstly, utilize the prebiotic to protect the probiotic inside the gut, and secondly, to overexpress the enzymes in order to overproduce the SCFAs. The prebiotic we chose is alginate, a carbohydrate suitable for site-specific degradation by colonic bacteria in humans and animals. Also, alginate can form alginate beads under certain conditions, which enables us to microencapsulate our engineered bacteria. We created and tested the durability and quality of those beads and showed that the beads did not dissolve after exposure into acidic conditions, simulating the gastric juice.
In parallel, we focused on the overproduction of three of the most abundant SCFAs in the gut: acetate, propionate, and butyrate. For each one, we developed a genetic construct consisting of an engineered biosynthetic pathway which alters the metabolic profile of the bacteria and drives the carbon flux towards SCFA's production. We designed 3 probiotic strains with different capacities for SCFA's production. The system is thus personalized and delicate, as the number of bacteria in each one of our 3 products is the same, but the promoters inducing the production of the SCFA's anabolic enzymes have scalable dynamics. Next, we tested the overproduction of the SCFA's enzymes for acetate and propionate. As you can see, these 2 SDS page gels, some enzymes are overexpressed while some others are not. For our next iteration, we plan to test different combinations of promoters and ribosomal binding sites to achieve optimal overexpression for all enzymes. In order for the probiotic to overcome the metabolic strain from producing the SCFA's enzymes, we inserted a module that increases the carbon flux. This carbon is derived by the degradation of extracellular cellulose. In a healthy gut, about 50% of cellulose is degraded by natural fermentation in the colon, and the short-chain fatty acids are being produced, feeding the intestinal bacterial cells. As we were informed by medical experts, the more serious the gut dysbiosis, the less degradation takes place. This degradation is catalyzed by BGLX, a protein that we cloned into our bacteria, and we also fused with an heterologous signal peptide, improving the extracellular transportation. We tested the module by cultivating transformed and non-transformed bacteria in minimal medium with cellobiose, which is an analog to cellulose in the absence of glucose. Only the bacteria transformed with a BGLX containing the signal peptide have an exponential rate of growth in the first 24 hours of incubation. To ensure ameliorate safety, we designed 2 distinct kill switches. One that ensures bacterial death after defecation through an auxotrophy-based system, and a second one that is activated upon administration of an orthogonal molecule Q-mate that ensures colonization's termination.
As a team, our priority was to carefully listen to those in need and understand the problems they face. To ideate Amathia, we turned to a wide range of experts to address holistically diet-related NCDs, such as doctors, microbiome experts, nutritionists, mental health experts, chemical engineers, software developers, food technicians, health entrepreneurs, government and non-governmental organizations. Through our discussion with stakeholders, we optimized Amathia in 2 different ways. Even though our project's main core was human-based, we integrated society's needs and implemented our design to fit to the animal gut microbiome. We adopted the capsule's dimensions to fit utilization parameters for animal use, as well as changing the administration method of our symbiotic supplement. So, we assessed the problem of NCDs through the prism of the One Health approach. One key factor that we also took into consideration is mental health. The gut-brain axis is a well-established term, and through our partnership with Team Aalto Helsinki, we moved towards implementing counseling from mental health experts in our application system, as well as acknowledging the correlation between the gut microbiota and mental health disorders. We also considered informing gastroenterologists and their patients regarding mental health issues through 2 independent, comprehensive guides regarding the relationship between mental health and enteric disorders, all through our collaboration with psychiatrists. We gradually elaborated more and more on this issue, making it eventually our excellence in another area.
It is vital of importance bringing to life a research project by implementing and integrating it to the real world and the market. For that reason, we communicate our design and showcase the 3D printed model of our capsule to pharmaceutical and probiotic industries, aiming to receive valuable feedback. We hope that since the design and the blueprint of our capsule are designed and finalized, it could be eventually available for manufacturing. Regarding our probiotic supplement, after contacting with one of the key players in Greece of the dairy industries, we concluded on implementing those strains in the production line of sterilized yogurt. In that way, we make a symbiotic product that is aligned with the core values of Mediterranean diet and that is easily implemented to people's diet. We also managed to adjust our product in animals by integrating the reflections of field experts. On the other hand, aiming to make our toolkit accessible to everyone, our products will be widely available to local pharmacies, accompanied with well-documented guides for both experts and the users to ensure safe implementation. As far as developing our product from a business point of view, Dr. Koutsioulis, CEO of EnzyQuest, suggested us on developing a business plan using tools like SWOT and pest analysis. Moreover, we managed to participate in one of the greatest innovation and entrepreneurship competition in Greece, Falling Walls Lab Greece, where Amalthea managed to take the first place among all the other competitors.
Throughout this iGEM season, we've worked with numerous teams on numerous subjects, aiming to exchange ideas and share our values. One of our most extensive collaborations was with Team Aalto Helsinki from Finland. They inspired us to acknowledge the impact the gut microbiome can have on your mental health. Together, we've co-evolved our two projects in ways such as ensuring the encapsulation method of our detection systems, safety mechanisms, and understanding mental health issues and act upon them through our human practices. Overall, we had a very good friendship and cooperation. Together, we hosted Microbiocosmos, an initiative aiming to invite other iGEM teams working on the microbiome to promote the importance of microbiome research, real-world implications, ethical considerations, and overall the implementation of our projects. Our fruitful conversation is summarized in a foresight report which aims to inspire future iGEM teams working on microbiome. Regardless of our own efforts, we have to acknowledge efforts led by other teams to ensure and fulfill the competition's goals. We've taken part in numerous collaboration efforts. All of those helped us understand abstract ideas such as the meaning and ensuring sustainability, promoting education for all, and understanding our work and effort. As for our engagement with the general public, one of our most important educational initiatives was introducing synthetic biology to the Greek high school curriculum. We prepared a set of educational materials for students and high school teachers and invited them in a seminar exclusively for them to receive feedback and to inspire them to start the very first Greek iGEM high school team. We finished this project by presenting those educational materials in a high school setting to the students face-to-face. This project also helped educate our ambassador team and gave them a taste to how to actualize a public engagement and educational activity. Amalthea is a complete prevention toolkit to tackle the rising threat of diet-related NCDs. We detect, evaluate, and ameliorate gut dysbiosis, highlighting the importance the gut microbiome plays in those disorders. So, it's time to finally listen to your gut. Are you ready?